So we're partners, and we also jointly run our lab here at Cornell. We have a pretty big lab. We have 3 permanent PhD-level research scientists, about 10 postdocs, 10 grad students, about 20 undergraduate researchers, many of them from engineering, but drawn broadly from the university. Today we're going to tell you a bit about 2 major projects in the lab having to do with what causes reductions in blood flow, either to the brain or to the heart, in models of neurodegenerative disease or heart failure, which sounds like 2 totally different things, and we are different people. But I hope we're going to show you that there's some common mechanism that's playing a role between these 2 things, and that there may even be some convergent biology here, which is hopeful. So, broadly speaking, in my and Nozomi's lab, we try to understand how different kinds of cells interact with each other to give rise to symptoms of disease in mouse models of neurological and cardiovascular disease. So what we want to know is which cells interacting with which other cells give rise to specific symptoms of disease. And we'll show you an example here today where a neutrophil, that's a class of white blood cell, interacting with an endothelial cell, those are the cells that line the inside of your blood vessels, that interaction is going to cause reduced brain blood flow that messes up cognitive function and reduced heart blood flow that messes up heart function. But to do this kind of work, we want to actually look at how individual cells are behaving inside a blood vessel or inside a brain or inside a heart. And we don't have reduced preparations where we can just grow something that is so much like a heart in a dish that we can study it and answer the kind of questions we're after. So all of these experiments are done in live mice. And in order to be able to see individual cells in live mice, we have to build some fancy imaging tools. And Nozomi and I are actually trained as physicists, and a good fraction of what our lab does is developing these new optics-based tools for looking at the behavior of cells inside live animals. So today there's going to be kind of 2 parallel stories about the causes and the consequences of blood flow reductions in the heart and the role that plays in heart failure, and then in the brain and the role that plays in neurodegenerative disease. And we'll talk about how a interaction between an inflammatory cell, a neutrophil, that's circulating in your blood, and the microvasculature, the cells that line the very small blood vessels in your body, what are called capillaries. We'll show how interactions between those inflammatory cells and those microvascular cells contribute to these flow deficits, and we'll show some pretty remarkable improvements when we block this process.
So, as Chris said, we are, I like to say, recovering physicists. It's, the jump between engineers and physicists it shouldn't be that big, but it is culturally kind of big. At any rate, so our lab actually develops a lot of the tools. I'm going to use the pointer on the screen. As our physics background actually allowed us to work on the microscopy itself. So we work a little bit on the technical side and develop some of the imaging techniques. And that is 2 and now 3 photon microscopy. This is a microscopy that was invented at Cornell by Walt Webb in applied and engineering physics, but it's now become a biological tool that's become very popular around neuroscientists. Our lab has been not just using this in the brain, but also developing the technology and working on the microscopy engineering side to be able to do this in lots of different organs. And so it requires that we actually work on the surgical side as well to be able to put literal windows into animals in different places. But then it allows us to look at the behavior of cells as if as if we're watching a nature program and looking at cells as they interact and do their daily business. They actually move, they interact, they fight, they sometimes cooperate. And so we today are going to talk about the brain and the heart a little bit.
Great. So let's talk a little bit about Alzheimer's disease. I really need to change this title. Alzheimer's disease has been with us for 100 years, but I don't think it's going to be the pandemic of the 21st century. We just had that. Maybe it's the scourge of the 21st century. So this is a photograph of Auguste Dieter. This is the first patient who was described as having Alzheimer's disease by Dr. Alzheimer himself. The paper was over 100 years ago, back in 1907. And just a couple of things to note. So in addition to kind of looking dejected here, around the time this photo was taken, Ms. Dieter was asked to write her name. And this paper kind of in the lower left of the photo is her attempts to write her name. And it took 3 tries, but she was eventually able to get Auguste. You can see, I can't tell where this is. Anyway, Auguste. And then she got D and couldn't remember the rest of her name. So that's what Alzheimer's disease strips from you. Really strips away the ability of your brain to form and store and recall memory. One of the first symptoms of someone who's developing Alzheimer's disease that you'll notice is you'll be having a conversation with them that feels quite normal. But then they'll ask a question that they asked just a few minutes ago and that you answered clearly. And then the reason is that the part of the brain that can take that experience of asking a question, hearing the response, and convert that into a memory, that's a hippocampal part of the brain, that part of the brain is just broken. And so the ability to form new memories is basically degraded. Over time, you start to lose existing memories, usually in a retrograde fashion. The last thing to go tends to be memories of songs that you enjoyed as a teenager. So for those of us that grew up in the 80s, it's going to be awesome. That was a really good decade for music.
No.
Feel sorry for the youngsters these days. It's not much to look forward to remembering when you're old. This disease is, in addition to its impact on people, one, it's quite prevalent. So 1 in 8 Americans over 65 has Alzheimer's disease. If you make it to 85, half of you, like it's 50-50, you're going to have Alzheimer's disease. In addition to the impact on people who have it, there's folks with Alzheimer's disease require a lot of care. And so there can be physiological and psychological impacts on caregivers. And this is very expensive. This will bankrupt Medicare by 2050 unless we have a new plan or we change Medicare taxes quite a bit. We spent over $280 billion on direct just care to Alzheimer's disease patients in 2018, and this is just growing. There have been a couple of drugs that have come onto market recently that show a little bit of mitigation of some aspects of disease processes and symptoms. But overall, this is not a condition for which there's particularly good drugs to be able to deal with it right now. So, I told you earlier that the kind of approach that Mayim Nozomi's lab takes is to try to identify particular symptoms of conditions and then identify the cellular interactions that cause those symptoms. In Alzheimer's disease, the symptom we're trying to understand is the fact that Alzheimer patients have about a 30% reduction in blood flow to their brain. Now that's a lot. You know when you're laying down and you stand up too quick and feel dizzy for a second? That's about a 30% reduction in blood flow. In that case, just for a couple of seconds, and you can already feel a cognitive symptom, right? You feel dizzy. So Alzheimer's patients, they have that kind of reduction to blood flow all the time. And it's likely that that contributes to the memory and cognitive problems of the disease. We didn't discover this reduction in blood flow. People have known about it for 50 years. But the mechanism that causes it has been unclear. If you don't know what causes something, it's really hard to treat it. The other thing I'll say is that this is an early feature of the disease. In retrospective studies, this is the first thing you can identify in someone who later goes on to develop Alzheimer's disease. So it's not like at the very end stage of Alzheimer's disease, you have reduced brain blood flow. So if we fixed it, like there's just a modest gain. This is a very early feature of the disease. If we could figure out what causes this and fix it, it could potentially alter the trajectory of the disease, improve folks' cognition as the disease develops. One thing I'll say, though, is this is going after a symptomatic treatment. We're trying to fix the reduced brain blood flow. It's not trying to go after causes. And I think that'll likely be a theme going forward in a lot of these very complicated diseases.
All right, so Chris told you about how horrible Alzheimer's is, and it really is. But I'm going to tell you about the heart, and it's not a contest, but actually cardiac and cardiovascular diseases does affect more people. And that's what a horrible thing to think about. But the truth is that most people are probably going to be affected by multiple diseases as well, and there's quite a bit of interaction in between these diseases. But these are just graphs to remind you that the heart is still quite important, and in terms of numbers, it actually does affect more people. But we'll also talk a little bit about the links in between cardiovascular and brain diseases. So we are going to focus our talk today about a specific kind of heart disease. And there's basically two kinds of heart failure. And what heart failure means is that your cardiovascular system and your heart cannot deliver enough blood to the rest of the body. But there's sort of two flavors. And one that's been studied a little bit for a longer time is called heart failure with reduced ejection fraction, which means that the amount of blood that the heart can push out is actually reduced. And so that makes sense that it affects the body because you're not actually pushing blood into the rest of the body efficiently anymore. There's a second form, which is almost as many patients or maybe even more patients, and it affects women a little bit more than it does men. And it's called heart failure with preserved ejection fraction. And in this form of heart failure, the heart can still pump, it can still squeeze and push blood out, but it actually can't relax very well, and so it can't fill up. And so there's still a shortage of blood that's delivered to the body. The first form, the reduced ejection fraction, there's actually quite a few drugs that are out there that are used for symptom management. However, with heart failure for preserved ejection fraction, there's only recently been the discovery of one class of drugs, and that can help with the symptoms in this disease. And it's kind of interesting because those drugs, they're sort of accidentally found. It's actually a diabetes and a kidney drug, and it just happens to work very well for heart failure with preserved ejection fraction. We don't quite understand why. So there's a big need for understanding what's going on in all of these diseases. One thing that's similar in between the diseases of the brain that Chris was talking about, so the Alzheimer's disease and the cardiac disease, is that there is a lack of blood flow. So usually when we think about the heart, we're thinking about it delivering blood to the rest of the body. But the heart itself has to support itself, right? In these forms of heart failure, one of the unexplained symptoms is that there's not enough blood within the heart wall and the muscle itself. And so physicians didn't really know the cause, and so they invented a clever acronym to cover that. So INOCA is ischemia, so lack of blood, and no obstructive coronary artery disease. So what that means is that there's a lack of blood, but they can't figure out, know what the cause is, so that the cause is not there. So INOCA is a condition that is well recognized, but it is actually an admission that we don't actually know the cause of the problem of blood flow to the heart itself.
So, I'm sure folks have heard enough about broken pumps. Let's turn back to interesting science, the brain. So one thing you might ask is, we've talked about how there's reduced blood flow to the brain in neurodegenerative disease, reduced blood flow to the heart in heart failure. Why have people waited so long to figure this out? Why have we tried to figure out what's going on? This has been 50 years, folks have known there's reduced blood flow to the brain. Why hasn't this worked out? Why isn't there already treatment strategies? And I think the answer has to do with the nature of the blood flow networks that we want to look at. So this is a cross-section of just the cortex, the outer kind of millimeter layer of the cortex of a rat, where blood vessels have been filled with a black ink. And you can see it's this beautiful, super complicated, kind of spaghetti-like network. And the problem that's leading to reduced blood flow could be occurring anywhere in that vascular hierarchy. It could be in some of the arteries or arterioles that bring blood into that tissue. It could be in those capillary beds that you see as that sort of really dense network, or it could be in some of these bigger vessels that drain blood back out of the brain. And prior to our work here, there had been efforts, the ways that people had to look at blood flow could either look at blood flow in a really big blood vessel, like the carotid artery in your neck. And when they did that, they would say, well, carotid artery looks normal, but blood flow is slower. Or they could look at the average blood flow to some block of tissue in your brain, which integrates over the flow in thousands to tens of thousands of little blood vessels. And when they did that, they said, oh, what's called perfusion, that perfusion is lower. But again, no sign as to what's causing it. And the reason is that you need some technique that allows you to go look individually at each of these little blood vessels. Look at the small arteries, look at the arterioles, look at the capillaries, the venules, the veins. So we need to look at all classes of the vascular hierarchy, sort of one blood vessel at a time. But as you know, if you just look at your finger, even if you put your finger under a microscope, you can't see all those tiny blood vessels. And that's due to the fact that the tissue is optically scattering. That's why we're not transparent, because light scatters as it goes through us. And that makes it hard to see those individual vessels. So this is where an imaging technique called two-photon excited fluorescence microscopy, which everyone at Cornell should know, two-photon excited fluorescence microscopy. It'll be on the quiz. This is an imaging technique that was invented in Watt Webb's lab and applied into engineering physics in the early 1990s. It is one of the most impactful scientific discoveries from Cornell. This has become the technique of choice globally for high-resolution imaging inside scattering samples. It's one of Cornell's most valuable patents. There's tens of thousands of these microscopes installed around the world, used for everything from basic biomedical research to preclinical drug testing. There are aspects of this that are moving forward as clinical diagnostic tools. What two-photon microscopy does is it makes it where, so we use a fluorescent dye to label cells that we want to see, and two-photon microscopy restricts the volume where we excite a fluorescent dye to one small three-dimensionally localized spot. What that does, it eliminates the blurring effect of optical scattering. So if I excite fluorescence in only one spot inside of a sample, it doesn't matter how much that fluorescence scatters on the way back to my detector. I still know it came from that spot because I only excited it at one spot. Now that's not an image. That's how much fluorescence I get from one spot. So to build an image, I would then take that laser spot and scan it from point to point to point inside the sample. And at each location, I measure how much fluorescence I get. And I can scan about 1 million points per second. And that's enough to take 3 or 4 frames per second. So it's like 3 or 4 frames per second, but now no blurring due to optical scattering and the ability to look down in 3 dimensions. So here we go, stepping down into the brain. Those were some of the bigger blood vessels up on the brain surface. Now we're looking at individual capillaries, the smallest blood vessels in the brain. And if you look carefully, you'll notice that there's what looks like little black dots that are zipping around inside those blood vessels. Anybody have an idea what those might be?
Blood cells?
Blood cells. Very good. Yeah. So your blood, it's about half liquid and about half made up of cells, mostly red blood cells. And the fluorescent dye we inject, it labels the liquid part of the blood plasma, but it doesn't label the blood cells. So the blood cells show up as little shadows. And actually by tracking the motion of those shadows, we'll be able to measure blood flow speed in individual vessels. So with these tools now, we have the capability to look into the brain of a mouse and to see the vasculature at all levels of the vascular hierarchy, arterioles, capillaries, to venules, and to quantify the blood flow speed in all of those vessels. So this is the tool we need to now go look and try to figure out why is blood flow messed up in the brain of a mouse that's been genetically engineered to get Alzheimer's disease.
Okay, to be fair, the brain was the first place that multiphoton, two-photon microscopy was applied to, and that's because the imaging is pretty easy. So where the real microscopists go now is actually in the heart. And so this is just an image to remind you that the heart is it's its own pump, so it is moving. And while there have been lots of studies in the past that have done things like paralyze the heart, taken it out, like cut it up or whatever, to be able to study its full function and then also its own delivery of blood into itself, you actually have to have it alive, pumping, and intact. And so it shows you how much harder it is to image the heart than the brain. So on the left side here, those are images that we get, and the red is the dye in the vasculature. So even though that we have this stabilization, there's still a lot of motion, so you don't get those same nice images that you get in the brain. We went to a faster scanning microscope, which is on the right, which gives you a little bit of stability, but it's still not quite enough to get fully stable images. And so we actually use a lot of computational tools to then reconstruct the images at different phases in the heart cycle. So the heart beats repeatedly. Actually, the breathing is also repetitive. And so we can actually go back and reconstruct each image line by line from where it happened relative to the heart cycle. And so after all that, you can get these 3D images, these reconstructions that show you what the heart is doing in a heartbeat. And so the image on the right is just cycling through the heartbeat. And so the big pink thing is a blood vessel. And the green is actually some of the cardiomyocytes all around that. And so this is one of, I think, the few microscopy images there are that actually capture at this scale of these tiny microvessels what's happening with the actual blood vessels that's perfused with real blood.
Well, here's microvessels in the brain. You can do it there too. I mean, I agree, it's kind of cool to do it in a moving heart, but this is doing the same thing as Zoe was just showing, but now looking in the brain of a mouse with Alzheimer's disease. The red shows blood vessels. The green shows those amyloid plaques. So, Kaoli, you've heard one of the things people look for is a characteristic pathological hallmark of Alzheimer's disease is the accumulation of amyloid or senile plaques. So those amyloid plaques in this mouse are labeled with another fluorescent dye. So basically our job, remember, we're trying to understand what causes reduced brain blood flow in Alzheimer's disease. These mice show that same reduced brain blood flow. And so we're trying to look at different levels of the vascular hierarchy to see where things are messed up. So we started out looking at some of these larger blood vessels like you see here, the surface arterioles and venules. And there's no difference between the Alzheimer's disease and the wild-type mice. So this isn't where the problem is. It's not in larger blood vessels. When we looked at the smallest blood vessels, though, individual brain capillaries, and remember, in capillaries, your blood cells flow single file. They're sort of all just lined up, one after the next. So there, we found an increased incidence of what we call stalled capillaries. So here on the left, this is like a film strip, where we're seeing the same blood vessel over about a second and a half. And this one on the left, you can see there's a little black object that's just staying in the same spot. So this blood vessel isn't flowing, called it stalled. While the one here, you could see there's some black spots from red blood cells, but from frame to frame, they're shifting in position because that blood vessel is flowing. So when we write papers, we have to talk about how many animals we killed in the conduct of the experiment. This analysis, I felt like we needed to write how many undergraduates we used up in doing this, because we were not able to develop automated techniques for scoring these individual vessels as flowing or stalled. And so for our initial work here, we had teams of undergraduates sitting and making this judgment capillary by capillary. First paper was about 250,000 individual capillaries that were scored as flowing or stalled by 3 different individuals, to keep it all above board. So anyway, but the result from that is we find here the fraction of capillaries that are stalled as a function of age for either Alzheimer's disease mice in purple or non-Alzheimer's disease mice in red. And you can see throughout the animal's age, there's an elevation in the number of these non-flowing capillaries. The other thing you might notice is that it's not very many. It's only 2% of capillaries that are blocked. So that means 98% are still flowing. And last time I was in college, 98% was still an A, right? That should be good enough. So I hope I'm going to convince you that that 2% of vessels being blocked, it has a very outsized impact on overall blood flow. And that has to do with the topology of the vascular network, basically how redundant that microvascular network is. One key question is what's causing these vessels to be blocked. So we basically developed labeling strategies to distinguish different possible causes. Things like a platelet aggregate or a clot, or just a red blood cell getting stuck there for some reason, or a white blood cell getting stuck there. Based on this, we determined that about 2/3 of these stalls were caused by a white blood cell, and we later determined it was a particular class of white blood cell called a neutrophil. These stalls are not permanent. They don't stay there forever. They have a half-life of just a couple of minutes. But it turns out that the vessels that stall once, they're more likely to stall again. So it means there's something wrong with that vessel, something sick about it, and it keeps stalling. I can still remember the day this happened. Tori Hughes was an undergraduate student. We were trying to figure out what kind of white blood cell was sticking in these blood vessels. And we knew it wasn't going to be a neutrophil, but we had to rule that out. So we gave the new undergraduate the task of ruling out neutrophils. And the way you did this was by injecting an antibody, a molecule that will bind to a specific protein on the surface of a neutrophil. And that antibody had a fluorescent label on it. So Tori forgot to dilute the antibody. So we're supposed to take the antibody and dilute it like 1 part in 10,000. So she skipped that step of the protocol and then injected this thing at full concentration into the mouse. And then she came and told us, like, oh, yeah, the experiment didn't work. And I was like, well, what do you mean it didn't work? She said, well, when I injected the antibody, it didn't label the stalls. It made them all go away. So she serendipitously discovered, for mechanisms that we still do not understand why, that a high dose of an antibody against this neutrophil-specific surface protein called Ly6G will lead to those neutrophils that were stuck in capillaries to no longer be stuck. It's a completely serendipitous discovery. We still do not understand why. So, but what this did do is it gave us a tool that allows us to manipulate this phenomenon. We even wrote serendipitously in the paper. We serendipitous, because otherwise I was like, how are we going to explain why this works? We just said we serendipitously discovered. But this gives us a tool to manipulate this phenomenon. So this is the fraction of capillaries with stalled blood flow after we give a drug divided by before. So here's a control drug. It's a 1. That means there was no change. Same number of vessels stalled before. And after, when we give this anti-Ly6G, we get rid of about 2/3 of the vessels that were stalled. You'll recall about 2/3 of the stalls were associated with a white blood cell. So we basically get rid of all of those white blood cell stalls. Now, when we did that, we then went and looked to see, well, what impact does that have? And we do this by measuring the blood flow speed by tracking the motion of blood cells in individual blood vessels. So here we have the volume of blood flow going into some arterioles that feed the brain before and after we get this drug. And you can see many of these vessels have sped up by several tens of percent. Looking in aggregate across lots of vessels. So this is a plot of the blood flow speed after we give the drug divided by before. So 0 would mean flow stopped, 1 no change, 2 it doubled. For a wild type mouse that gets the real drug, there's no change in blood flow speed because they don't have stalls and there's no stalls to get rid of. An Alzheimer's disease mouse that gets the control drug, has no change in speed because that doesn't modulate the stalls. While an Alzheimer's disease mouse, either young or old, that gets the real drug, we get a median blood flow speed increase of about 30%, making up the deficit that these Alzheimer's disease mice have relative to wild types. So at this point, I hope I've convinced you that by looking in detail at what's happening to blood flow in a vessel-by-vessel approach, we were able to identify this mechanism, this cellular mechanism, where white blood cells are getting stuck in capillary segments. And we serendipitously discovered, thanks to Tori Muse, who just finished her PhD, by the way, at University of Copenhagen, I think.
so.
Yeah, University of Copenhagen. So thanks to the lucky fingers of Tori Muse, we discovered a drug that allows us to block that stalling phenomena, and then showed that when we do that, blood flow speed increases. So this is evidence that we've identified the cellular mechanism of decreased brain blood flow, at least in these mouse models of Alzheimer's disease.
Okay, so let's switch back to the heart for a minute. We wanted to study these models of heart failure. And so the way that we give heart failure to mice is we actually feed them a high-fat diet for several months, and then also increase their blood pressure. And so this was actually a recently developed model.
Folks, there's a lesson in that. In case you didn't hear it, like.
Yes, absolutely. Okay, so when we subject these mice to the high-fat diet, which I think they like a lot, unfortunately, what you actually get is an enlargement of the heart. And so the picture on the left shows the heart of a normal chow-fed mice, just a normal chow, so a normal mouse, and then a high-fat diet plus the high blood pressure mouse. And so you can see that it's quite a bit enlarged. And the picture with the green, that shows the outline of the cross-section of the muscle cells. So you can see that the cells themselves have gotten quite large. These images also sort of give you a hint of why the focus of maybe a lot of these heart failure studies was not so much on blood flow for a long time. It was on what is it that's driving this large enlargement of the cells, and because it's quite obvious. And so that's just some quantification, but hopefully the images are convincing enough. So we turned our imaging tool to looking at what's going on here, so in these windows. And so the top shows the blood vessels within the beating heart of a normal mouse, so the control mouse. And so on the on the left, it's the unstimulated heart, and then we actually give epinephrine to increase the heartbeat and increase the squeezing of the heart. And so you can sort of see the very complex motion of the heart. On the bottom, these are the animals with heart failure. You can see that there's still motion and there's still some response, but it's not as it's probably not as much as you would get in the normal heart. And now, I swear that we weren't trying to copycat the brain people, but it actually turns out, and I was not expecting this because I was really, brain heart, it should be very different. It turns out that if we look very carefully, you can actually see the same phenomenon in these heart vessels that you can in the brain. And this was such a surprise because I really wouldn't have thought that these were similar in any way. But in the images on the top, what you can see now in red is that same cell that was implicated in the Alzheimer's disease, the neutrophil. Normally what it does is just flows right through all the blood vessels very quickly. But what we found when we used our microscopy to look in the mice with heart failure was actually the exact same thing that Tori had discovered in the Alzheimer's mice. So what happens is that every once in a while, one of these neutrophils actually gets stuck in the capillaries, just like it does in the brain. And so it seems like what we're seeing is an increase in the same kind of phenomenon, the number of stalled vessels that are actually blocked by, coincidentally, the exact same cell that we found in Alzheimer's disease. So we did a lot of quantification, which I'll sort of skip over, but in the end, it looks like it's very, very similar to Alzheimer's disease in the sense that there are just a few capillaries, a minority of capillaries that are plugged, and it's the exact same cell. So that was a big surprise.
Great. So in two different mouse models of major conditions, neurodegenerative disease and heart failure, we found that reduced blood flow to that orbit, either the brain or the heart, is caused by occlusions at the capillary level by these arrested white blood cells. And in the case of the brain, we figured out how to fix it. So we're a step ahead. But does it matter? Let's suppose we fix this, we improve brain blood flow, and nothing changes about the disease. Well, then probably that isn't a great therapy. So we wanted to know, what difference does this make for cognitive function and disease progression in these neurodegenerative disease mice? So first we did some cognitive testing to try to assess their memory function. And they can't take little surveys like the NSD exam or something. But it is kind of cool how you test the memory function in mice, so I thought I'd tell you how one of these tests work. So this is called the object displacement task, and it takes advantage of the fact that mice are curious little critters. They just like to explore the world around them. So what we do is we put them in kind of a boring box, about 2 feet on a side, and it's just like plain white walls. They're not padded, but it's like you're sort of getting the idea. And there's nothing in there interesting except 2 little toys, like 2 little chestnuts. And so what the mice will do is they'll spend a lot of their time like exploring and checking out those toys. And then we take them out of the box and like go put them back in their home cage. They had lunch or whatever. And then after lunch, we grab them and we throw them back into the box, except when we put them back in the box, one of the little chessmen is moved to a new location. Now let's suppose some sadistic Cornell professor did this to you. What do you think you would do when you went back in the padded room after lunch?
You'd spend more time looking at the one that got moved. Like you'd be wondering why he was over here.
Right. So the one that moved, so you would do this. The one that moved, you would find you'd spend more of your time looking at it. Why? Because it's kind of cool. It's different. It moved. It's more interesting than the other one. So mice will do the same thing. They'll spend more of their time looking at the object that moved, unless they don't remember the first time they were in the box, right? Then it's a brand new day. There's 2 new chessmen, and they're equally interesting to explore. So what we do is we track the motion. So this is tracking the position of the mouse's nose as they're running around in a box. Then we quantify how much time did they spend looking at this object as compared to this object. So the data I'll show here is a lot of groups, don't worry about it. So 50% here means equal time looking at both objects. And here are mice that spent like 60-75% of their time looking at the moved object. So first I want to compare the 2 groups with the red arrows. That's an Alzheimer's disease mouse group at baseline and wild-type mice at baseline. So at baseline, the Alzheimer's disease mice show no indication of memory in this task. It's equal time exploring both objects, while the wild-type mice, it's about 65-70% of their time looking at the moved object. The most exciting set of groups to examine here are these 2. This is the same group of 10 Alzheimer's disease mice at baseline and just 3 hours after a single injection of this drug to improve brain blood flow. It's the most rapid recovery of memory function ever recorded in an Alzheimer's disease mouse. It's not like you get a prize for that or something, but it is the most rapid recovery of memory function folks have described. This was in mice that were just 10 months old. For perspective here, these Alzheimer's disease mice, they first start to show signs of cognitive decline around 8 or 9 months, and then they'll live till about 2 years. So they have kind of a long, extended phase of cognitive decline. Here we did a set of experiments where we basically let these mice age out from 10 months to a couple of years old. And every couple of months, we gave them an injection of either the real drug or the control drug. And every time the pattern looks like a little B, that means that we were able to improve the function with the real drug. So out to mice as old as 17, 18 months of age, a single injection of this drug improves cerebral blood flow, and that recovers memory function. Older than that, when we inject the drug, we still increase brain blood flow. But increasing brain blood flow is no longer enough to rescue the memory function in the mice. So at that stage of disease progression, like other aspects of what's going on to damage the brain in Alzheimer's disease have gotten to the point where improving blood flow is no longer enough to rescue memory function. So just to kind of summarize the story with Alzheimer's disease. So about 2% of capillaries were blocked by an arrested white blood cell. when we interfered with that arrest of the white blood cells, blood flow came up by about 30%, and that was correlated with a dramatic and rapid improvement in memory function in these mice.
Okay. All right, so it worked in brain, and I swear I'm not copycatting, I'm really not, but we might as well try it in heart as well. We found it was the same cell, and so we actually did decide to use the same strategy, the same treatment of the same protein in the heart, just to actually see what happens.
Copycat.
Well, okay, so we don't have the same kind of assay. We don't ask cognitive tests of mice in the heart failure models, but we do have another assay that we could do pretty quickly. And so the heart beats, right? And so it pushes blood, but it also has to relax to fill back in, and so it can prepare itself for its next beat. And that's called diastolic function, the idea that it can relax. And it's actually one of the things that is reduced in this heart failure model. And so what we did in this experiment was treat with the same drug, actually for quite a while. And then what we found was that the heart actually can relax better after treatment for, in this case, it was a whole month. And so that's very exciting because there aren't actually, for this kind of heart failure, very many drugs that have been implicated. And this was actually a drug that was improving the function through treating this interesting and novel mechanism that's also shared in the brain. Actually, with a month's treatment, you can actually get changes in some of the scar tissue that forms in the heart, so the fibrosis, which is also very encouraging. In the heart, we were also able to measure how much oxygenation there is, and this is actually through a probe that you inject, and it binds to tissue that has low oxygen. And so low oxygen tissue gets very bright, and it turns out in the heart failure models that you can, with this method, measure that there is a decrease in the oxygenation, so the tissue is suffering from a lack of oxygenation. And then treating these neutrophils actually improved that oxygenation after about a month of treatment. However, based on the idea that in the Alzheimer's situation, the cognitive function of the mice was actually able to improve very quickly, presumably just by increasing the blood flow, we actually decided to test this in the cardiac model as well. And it turns out that, these are still ongoing experiments, but it turns out there is actually something very similar. So in the heart, we treated these animals for just 1 day, which is not long enough for cells to remodel or anything like that. And it does turn out that you can improve the oxygenation in just 1 day. And so that was very interesting because that means, or that suggests, that this same mechanism where neutrophil stalls are decreasing blood flow and then therefore probably decreasing the oxygen in the Alzheimer's case in the brain is probably happening in the heart. And very excitingly, you can see a very, very fast change that would that indicates improvement. So just like in Alzheimer's, what we're really concerned about is how the patients really feel. In the case of the cardiac studies, we actually do an exercise test, inspired again by the rapid turnaround of the cognitive function in the Alzheimer's mice, we did this, the test where we actually did an exercise test with a rapid treatment. And it is actually showing some trends that even with just 1 day of treatment, you can actually get improved exercise tolerance in these animals. So I think things are very interesting because we're seeing in 2 totally different body parts, 2 totally different diseases, some similarities, and got insight from looking at the brain on the heart, and we'll see whether we get any insights from the heart that goes back to the brain, but we'll see. All right, So in summary, what we found was actually the exact same things in the heart that we did find in the brain, and that was enabled by these imaging techniques where we can see at the single vessel level. So we were able to see in both cases that there are cells that are blocking up the vasculature, the very tiniest vasculature, the capillary bed. And then we were able to use the same drug that we had discovered in the brain case and use that in the heart case, and then see the exact same sort of very rapid recovery and function. And so this actually leads us to a couple final thoughts. So it turns out, and it makes sense if we think about this, sort of take a step back. It turns out that in both of the diseases, we're seeing the same phenomenon. And perhaps it makes sense when you consider that both of the diseases are lingering diseases that are chronic. And so patients develop these over many years, and there's no like acute event that happens, but it's sort of a slow buildup of problems. And so in both of these cases, these are diseases where you have inflammation that is present for very long periods of time. And so it makes sense that perhaps in both of these diseases, you're getting the same phenomenon. But I think it wasn't appreciated that there are some very small changes at the capillary level, and both in terms of the number of capillaries that are affected and the size of the blood vessels that are affected. But they have a large or fairly large impact on blood flow in both of these organs. And both of these is enough that it actually impacts organ function. So actually the thinking processes, the cognitive and memory processes, and then the blood flow delivery in the heart that actually makes a difference for hypoxia. Okay, so in both cases, we could rescue blood flow and then get a functional change. And this really suggests sort of a completely different way of thinking about the impact of these diseases. Now, in both cases, we're not dealing with the fundamental causes of the diseases. In the Alzheimer's models, it's a genetic cause in that particular model. It's still high fat, high blood pressure in the mouse, in the heart failure model. But we were able to treat the symptoms that maybe matter the most. We're not changing the absolute causes, but maybe we're making the symptoms that the patients really care about, how they feel, a little bit better. So there's some hope. I do have to make the caveat that this drug is not something that is translatable. It is something that is only in mice right now. It is a protein that is a mouse-specific protein. And we are working on things like how to translate to other compounds that might be usable in humans, and testing that in both different diseases. So with that, I'd like to acknowledge that this has been the work of very many people, all those undergrads that we used up, and actually now we're starting to use up more undergrads in the cardiac case as well. But it's really been work of a huge group of people, graduate students, postdocs, and scientists at all different levels. And with that, I think I'd like to thank you for coming to this. Thank you.